Today, the Professor Annalena Venneri, Dr. William Sally Luis, Dr. Spallazzi Marco, and the Pharm.D, Professor Andrea Spini. And we have as a moderator today, Dr. Manca Riccardo.
Thank you very much. I think I will skip to this. Thank you all for staying behind. I know it's more or less lunchtime, so probably our stomachs are grumbling right now, but please bear with me, bear with us for the next hour. I think it's going to be interesting. So for this roundtable, we organize it in a sort of 2 hours, more or less. We will start with a series of presentations to introduce you to some hot topics, because at the moment, for those of you that maybe know a little bit more about Alzheimer's disease, it's quite an interesting moment in time, because there are many developments in research for new medications as well. We're going to focus on a specific topic, that is the role of A-beta, so beta amyloid accumulations in Alzheimer's disease. Also in light of some recent scandals, let's say, some problems in the scientific literature. And I will start actually introducing you all to a research case of research misconduct. And then the other colleagues will talk more about implications for research integrity, but also the implications for the clinical aspects for Alzheimer's disease. So what are we talking about? We're talking about publications that is from 2006 by this gentleman, Sylvain Lesnay. This guy is still a professor, as you can see, associate professor at Medical School, University of Minnesota. And in 2006, this paper became a very important paper. As you can see, it was published in Nature, that is a very important scientific journal. But as you can see here, the article has been retracted. Just briefly, what is the paper about? Luckily, it's not about clinical research, so there were no patients involved. It was a study on mice. But this was the very first, or one of the first studies that really found, let's say, some evidence that the accumulation of beta amyloid, in particular of a specific type, as you can see, A-beta star 56, was associated with impaired memory in mice. And also when this specific type of beta amyloid was translated into young rats, as a consequence, as a behavioral consequence, there was a reduction in memory performance. So the authors were quite happy about it. Of course, they proposed and they stated in their manuscript, okay, beta amyloid is actually implicated in memory impairment. So the type of memory decline that actually seems to mimic what we see in patients with Alzheimer's disease. So the paper was published in 2006, and it went on to become a very popular paper. As you can see, I've taken this number of citations from Scopus very recently. As you can see, it's been highly cited, and at the time, an investigation from the Science Journal started in 2022. It was said to be the 5th highest cited paper in Alzheimer's disease. So it has been quite influential in probably also shaping the field of Alzheimer's disease. Very briefly, what has been found in the past 2 years, evidence of image manipulation. So I'm not an expert about Western blots, but using also very novel tools based on AI. What other researchers in the field found is that this author, the first author of the paper, the guy that I showed you in the first slide, probably manipulated and probably duplicated some of the Western blot patterns to make the results more significant, probably. So in the past 2 years, there has been an investigation, but let's say most of the co-authors actually agreed to retract the paper, but the senior author, so Karen Ash, only agreed this year, so only a few months ago, only after another paper with a replication of the same findings has been published. So there was a lot of resistance to let it go. Of course, this sparked a lot of discussion in the scientific community at this stage in the past few years. Why? Probably also because starting in a few years ago, we started to have the first results for the trials, clinical trials that were used to test the first drugs to clear the amyloid plaques from the brains of patients. The results were not clear-cut, they were not fantastic, probably. The effects were quite small. So, of course, there was a fear in the scientific community that there might have been a bias in the literature, of course, because this paper and others in the field gave a lot of strength to the hypothesis that amyloid was really important. But of course, what we see today is that there is a mismatch between the amount of funding and efforts that have been put into studying amyloid and also anti-amyloid drugs, and the little effects that we have today and the availability of drugs, basically. And this was the case of research misconduct, and I will leave the floor to Sally to talk about research integrity a little bit more in general.
Now, first of all, I'd like to thank you for your kind invitation. I am delighted to be here with you today. First of all, I wish to invite you to read the European Code of Conduct for Research Integrity by all European academies. Now, in this document, there is a statement that is extremely important and that we must always keep in mind when we are faced with a suspected case of research misconduct. Anyone accused of research misconduct is presumed innocent until proven otherwise. In the case of the Alzheimer's disease paper that we are talking about today, the evidence of image manipulation was overwhelming, and therefore the paper was correctly retracted. Now, this chart was published in Nature at the end of December 2023. It displays the increase in number of retractions over the past decade. Roughly 50% of the retractions were due to issues with the authenticity of the data, and only 6% were due to genuine mistakes in the research process. In this chart, you can see that in 2023, there were over 10,000 retractions. It is generally acknowledged that the rewarding system depends on metrics such as the number of publications, the number of citations, the total amount of awarded grants. These metrics may encourage researchers to engage with research misconduct, fabrication, falsification, or plagiarism, and or other questionable research practices. The system, therefore, incentivizes quantity more than quality and novelty more than reliability, and fuels the so-called publish or perish mentality. But what is the cost? What is the impact of research misconduct? The cost of research misconduct is estimated at hundreds of millions to billions of dollars per year. Cost of fraudulent studies and the studies that follow to confirm irreproducible results. And also the cost of inquiries and investigations. But what concerns me is, and what I think is more dangerous and shameful, is the impact of research misconduct on patient safety and well-being when clinical trials are informed by fake data. Research misconduct can also have an impact on careers that are destroyed or tarnished. Now, so far, I have concentrated on violations to research integrity, but the lack of competence in research methodology is equally serious. In 1994, Doug Altman, I'm sure that you all know Doug Altman, he is a famous British statistician, wrote an editorial in the British Medical Journal entitled "The Scandal of Poor Medical Research." And what did he write? Huge sums of money are spent annually on research that is seriously flawed through the use of inappropriate designs, unrepresentative samples, small samples, incorrect methods of analysis, and faulty interpretation. We should be appalled. This is surely a scandal. Can I have some water, please? Excuse me. As the system encourages poor research, it is the system that should be changed. We need less research, better research, and research done for the right reasons. Abandoning using the number of publications as a measure of ability would be a start. Now, Doug Altman wrote this editorial in 1994, 30 years ago. But I'm sure that you will remember that during the COVID pandemic, we were in this exact situation. Nothing had changed. Many thousands of studies were conducted, and they had absolutely no value because the sample size was too small or they were badly designed. In conclusion, all stakeholders, and by all stakeholders, I mean researchers, regulators, academic institutions, journals, academic societies, all stakeholders are responsible for promoting research integrity and research methodology. Research integrity and research methodology proceed hand in hand. How can we promote research integrity and research methodology? By focusing on education. There are a few Ph.D. students here in the audience who have followed my course on ethics and research integrity. By modifying the reward system, I'm sure that you are all aware of international initiatives such as DORA, the San Francisco Declaration on Research Assessment, and COARA, the Coalition for Advancing Research Assessment. By creating an environment and a culture that value critical thinking and open dialogue about the quality of research. We must concentrate on the quality of research. By regulating procedures for handling suspected cases of research misconduct. That's all.
Thank you, Sally. Now, Dr. Spallazzi will talk about some more clinical aspects of Alzheimer's disease.
Okay. Hey, hi everyone. Thank you, Riccardo, for the introduction, and thank you to all the scientific committee for inviting me to join this very interesting roundtable. It's also a pleasure to be here in front of such a young and motivated audience. As Riccardo pointed out, the retracted paper by Lesnay has historically reinforced the role of amyloid as the main driving force of Alzheimer's disease pathophysiology. But if that specific oligomer, A beta 56, is no longer considered particularly relevant, does the amyloid pathology and the amyloid hypothesis still stand as the most accepted interpretation for Alzheimer's disease pathophysiology? My aim today is to provide a very brief overview and some key concepts to help us navigate a very complex topic. So let's get started. As you may know, Alzheimer's disease is the most common cause of dementia, accounting for approximately 60 to 70% of all dementia cases. And you can understand how with the aging population, the projected rise in cases and the substantial lack of disease-modifying therapies presents an enormous challenge, not only for researchers and the healthcare system, but for the society as a whole. And we need to be mindful that even with a future game-changing therapy for AD, patient access and treatment costs will continue to pose a significant challenge for our healthcare system. Alzheimer's disease should be understood as a multifactorial disease, with age being the main risk factor. And our lifetime risk is mainly shaped by a complex interplay of direct, acquired, protective and risk factors, as well as polygenic predisposition. As you can see, there is a list of protective and risk factors. I'm not fully convinced about the marriage as a protective factor, but there's a potential point of discussion. No, I'm joking. And but if it's true that up to 40 to 50% of Alzheimer cases are preventable, or at least we can significantly delay the clinical onset, it's equally clear that Alzheimer's disease rests on a strong genetic foundation, mainly linked to APOE genotype. In fact, about 70% of all Alzheimer cases have at least one APOE4 allele. Given its great importance, let's delve a bit into Alzheimer's disease pathophysiology. Despite our significant advances in the understanding of the disease, we still face a number of uncertainties, and some gray areas remain. I don't want to sound paternalistic, but at this point I think it's crucial in Alzheimer's disease, and it's very important to maintain a rigorous and proper approach for Alzheimer's disease research. Moving on to neuropathological features, we have mainly three features: amyloid pathology, tau pathology, and neurodegeneration. And it's really important to know that the brain pathological changes start many years before the onset of clinical symptoms, and we can understand the challenge in addressing a disease that even in its early clinical phase has already a very high degree of neurodegeneration. Moreover, we are discovering that probably there are some other key mechanisms involved in the pathophysiology. The most relevant are probably neuroinflammation and vascular pathology. And it's interesting to note that the amount of beta amyloid does not correlate with cognitive decline, whereas the tau distribution and the tau accumulation closely mirror the cognitive symptoms, as you can see in these tau PET scans. So I mean, less tau less cognitive impairment, more tau, more severe cognitive decline. And to further complicate the puzzle, we still don't know if the different components of amyloid pathology, I mean oligomers and plaques, have a different and divergent role in the pathophysiology. So at the end, is amyloid a cause, an effect, or just an epiphenomenon? Probably to consider amyloid just an epiphenomenon is an excessive downscaling, but at the same time, we still don't know if the role of amyloid is causative and has a direct impact on neurodegeneration. Here you can see a table with some pros and cons for amyloid hypothesis. You can see that is a sort of neck and neck race, but to be honest, we don't have still the conclusive answer. And I'm going to conclude. We are experiencing a revolution in Alzheimer's disease, not yet therapeutic, unfortunately, but a revolution nonetheless. And I mean in the field of diagnosis and understanding of biological processes. Thanks to the development of in vivo biomarkers, we are now able to track the biological changes of the disease and to improve significantly our diagnostic accuracy. And until recently, we had CSF and PET biomarkers, but now also plasma biomarkers. Plasma biomarkers that have shown an impressive accuracy in terms of tracking biological processes and also in terms of diagnostic accuracy. So hopefully, we are moving toward personalized medicine that is able of capturing the great heterogeneity of the disease. Yeah, I'm done. Thank you for your attention, and with pleasure, I leave the floor to Andrea.
Thank you, everyone. Thank you for this kind of introduction, and thank you for the organizer for this invitation. So I will give you a brief overview about the drug approval process and pharmacotherapies authorized for the treatment of Alzheimer's disease. So as you can see, there is on average only 5 out of about 4,000 drugs studied in the laboratory go on clinical trials. And the current success rate of a drug candidate from the beginning of the clinical trial to receiving marketer approval is about 10 to 20%, and it has not changed during the past few decades. So from drug discovery phase to authorization of a drug can take 10 to 15 years. Phase 1 clinical trials are the one that give a brief introduction, the safety of the drug. Phase 2, the dose will be searched, and then the phase 3 clinical trials, comparative effectiveness is studied. So for drug development, it takes only a lot of there is a very expensive process because it can cost about $30 million. So, however, this is the regular drug development process. So phase 1, phase 2, and phase 3 trials. However, there are certain circumstances where this process can be shortened and marketing authorization can be given also on surrogate endpoints. And so there will be confirmatory studies on the market. So There are accelerated approval procedures to facilitate the development and expedite the review of drugs to treat a serious disease and fill a medical need. So the goal is to get important new drugs to patients sooner. So in this case, Alzheimer is Alzheimer disease. There are only drugs that improve symptoms, but we don't have. But these drugs do not have profound disease-modifying effects. So here you can see an overview of all the drugs that are approved for the Alzheimer disease. There are symptomatic drugs, for example, cholinesterase inhibitors that increase the amount of acetylcholine in the brain through the inhibition of cholinesterase enzyme, which breaks down the acetylcholine. There are the memantine, which is an antagonist of NMDA, which modulated the glutamate activity, which is another neurotransmitter implicated in the cognitive process. So these drugs treat the symptoms of the Alzheimer disease, but significantly and without significantly slowing down disease progression. So in the last 15 years, there have been a lot of research to find some therapies that try to modify the disease. So these therapies are monoclonal antibodies, which are not approved in Europe. So these therapies aim to impact the biological process that cause neurodegeneration in the Alzheimer disease, such as Marco previously said, the accumulation of beta amyloid plaque. So there is the aducanumab, lecanemab, and donanemab. Each of these drugs are approved by the FDA. All these drugs are approved through an accelerated process. So as we saw before, despite the aducanumab, the pharmaceutical company of aducanumab announced that the production and the commercialization of aducanumab stopped in November 2024. And the EMA, the European agency, stopped the production, refused the marketing authorization of aducanumab and lecanemab. So just to give you a brief overview, so these drugs are all monoclonal antibodies, but act on different pathway of the beta amyloid formation. So so each of these drugs can have different efficacy and safety profile. So then I leave the floor to Professor Venneri, which explain better the risk safety profile.
Thank you, Andrea, and thank you to the organizers for the kind invitation. Now, my job is more to approach the issue of how the clinical trials are designed, and especially what are the critical points in the evaluation of efficacy or treatments, or especially the most recent treatments of Alzheimer's disease. Now, the main point is on the choice of outcome measures. As we've seen, starting from from treatments that actually are meant to treat the symptoms. Bear in mind, Alzheimer's disease is primarily a cognitive disorder, so a disorder which sort of deprives us of our mental abilities. Well, the primary endpoint should be related to what the main symptoms of the disease are. In reality, what has happened is that with increasing sophistication of drug tested in this field, we have gone from using outcome measures which were of cognitive nature, so testing whether or not the drug was having an effect on the symptoms. We have gone to more qualitative measures. And indeed, the FDA approval or the recent drugs which are under evaluation in Europe or have been denied approval in Europe have used qualitative measures. Let's see in details what has actually happened. Well, for symptomatic treatments, the main outcome measure that FDA and other regulators would see as appropriate was the ADAS-Cog, which was the primary endpoint. There were secondary endpoints as well on brain atrophy and whatever, but unless there wasn't a positive effect on that, the drug would not get any further than the trials. However, for the recent treatments, what the approval has been based on is on a primary outcome, which is the clinical impression of change, or based on clinical impression of change, and the main outcome has been this the clinical dementia rate in sum of boxes. Then there were as well secondary outcomes which were not particularly brilliant, but the pain point was that. Let's have a look at what the difference between the two types of outcome measures are. Well, as you can see here, the ADAS-Cog, I've given you a summary, not listed all the tests, but does have a range of domains which test memory, attention, problem solving, and so on. Here in the CDR sum of boxes, what is actually required is for the patients or the carer to give their impression of how well the patient is doing after the start of the trial. And there are 6 fields that, with a Likert kind of scale. Now, I've taken here as an example the data from the lecanemab drug, which is the outcome of the Clarity AD trial. And as you can see, the difference between lecanemab-treated patients and placebo-treated patients was 1.1 decline in the treated group and 1.6 decline in the CDR sum of boxes in the placebo group. But I would like to draw your attention on what is clinically relevant. What we know is that on this scale, which, as I say, is primarily subjective, you need to have at least 1 point change for between placebo and control at the early stage of the disease, and 1.63 when the disease is more advanced at the mild stage. Now, one thing which we have to bear in mind is that there is no drug which doesn't have side effects. Here there is a list of the side effects which have been observed in the Clarity AD trial. And one of the most important ones are the effects on the brain. Brain swelling, edema, or brain hemorrhage is one of the most important factors. In some cases, this particular effects on the brain has led to catastrophic death. So here are some points for our discussion today. What are the problems which I see in evaluating treatment efficacy? As we've gone from symptomatic to so-called disease-modifying treatment, we seem to have gone from objective measures of cognition to subjective measure of changes. And as you can see, the main problem in this case is inflation of placebo effect, but also with the hormone drug, which is so inclined to side effects, the unblinding of patients and clinicians is pretty evident. The other thing which we need to focus on is on the meaning of the statistical significance. Is the statistical significance necessarily a clinical indication of a clinical benefit? The overemphasizing of findings when actually the difference between placebo and treated patients is very small. Objective benefit versus risk or side effects. This is something that we need to bear in mind. In addition, bear in mind that these particular forms of drugs are actually extremely expensive. They cost the health systems about $30,000 per year. And is, as you heard from Marco, the Alzheimer's disease is a very, very common condition in older age. Therefore, is that sustainable for any health system when the benefit is so small? But most of all, it is the topic of our roundtable today, is the integrity of the science behind these very expensive drugs. And we wonder whether this has been overinflated by trying to promote a theory behind the pathophysiology, which by no doubt, amyloid plays a role, but because it plays a role in aging in general, as Marco highlighted, a large amount of amyloid is found in elderly people with no cognitive complaints, but most importantly, a large amount of amyloid is found in everybody over the age of 80, which is an important factor here. And therefore, we need to discuss on whether or not the actual integrity of the science that underpins the amyloid hypothesis is something that pharmaceutical companies should still invest on. Thank you.
Thank you very much to all of our panelists for the presentations. And now we would like to open the floor to questions. So if you have any question, please raise your hand and somebody will come and hand over a mic to you. And don't be shy, please. Also, if we have questions from online, I understand that there are people following us remotely. Questions from the front row?
Why do you think we are in a situation where the FDA approves many more drugs than the EDA? If at the end of the day, they can both do the same test and they can both know the side effects.
I think we're gonna share the mic.
I can answer your question in the way I see it. There are major differences between the health system in the United States and the health systems in Europe. The majority of the I'll give you an example. As you know, I've worked for many years in the UK. The UK the equivalent of EMA in UK has approved lecanemab. However, the NICE, which is the sort of health system watchdog, has actually denied the drug on the NHS budget. So it's available as a private prescription, but it's not available on public money, because the evaluation was the risks are too high and the benefits are for a too small amount of patients, because you need to have special characteristics. You cannot be an ApoE epsilon 4 carrier. You don't have to be on blood thinning drugs and so on. So there are so many things that they didn't think it was worth the risk and it was worth public money spending. Anybody else?
Just one thing, because of course both FDA and EMA have a committee that evaluate the drugs before marketing authorization. So I think that the EMA committee said that there was not sufficient benefit according to the risk, just Professor Savennari said. I think that there was in the discussion of the committee of the FDA also some opinion that were in line with those expressed from the EMA, but the FDA on the basis of the fact that these drugs are approved only for specific population and on the difference of the healthcare systems, of course, approve these drugs in the US.
If I may add, that there is also another issue, which especially was the case in the first drug which was approved for aducanumab. There was an incredibly strong pressure from the Alzheimer's Association, which is extremely powerful in the United States, but also there was an incredible pressure from patients' organizations that were led to believe that this would be the solution to patients' problems. I do recall I was at the Alzheimer's Association conference when the aducanumab was approved. And what happened there was the actual chairman of the association, Maria Carrillo, who was saying, you know, we finally have the solution to, we are heading towards a world without Alzheimer's disease. It was very emphatic. You were there, Ricardo, you remember. But, you know, and we were almost watching her in disbelief, because they really wanted to believe that, but at the same time, you know, for the people like me who have been always extremely critical of this sort of acritical view about the role of amyloid, I can understand. The pharmaceutical companies have invested millions and millions on the pressure. And this is something which, you know, brings us back to what Sally was saying earlier. The pressure of some very influential people within the field of Alzheimer's disease who have shaped the field. And unfortunately, I must say, this is only one case that we have been discussing here, but there is a similar case with 132 papers found of one of these scientists which have fabricated data or you know, altered data. And this has had an influence, again, in Alzheimer's disease, but at the same time has had an influence in Parkinson's disease. I mean, the field of neurodegeneration, for the tragedy that it is for the patients, is unfortunately prone to this kind of problems.
Any more questions? Yeah, another one?
Hi. You opened the discussion talking about like the problem of faking data and publications, and I wanted to ask, like, do you think it's going to be possible in the future, like, to have more controlled way of the publications? Because for Alzheimer's, but also for other like very important diseases such as Parkinson or for HIV, there is like also a strong attention from like the normal people, and so like it can create really big damage not only in the scientific community but also in the population, because right now I think many people know about like the amyloid problem linked to Alzheimer's, and so many people maybe believe that like the solution to Alzheimer can be that easy, but actually is not. So what do you think about it?
I'm the moderator. I'm not supposed to answer questions.
Are you referring to the fabrication of data? Yes, yes. Well, at the moment, lots of companies are developing AI software to detect fabricated papers. You have probably heard about paper mills, businesses that produce fake papers and sell authorship. Well, in actual fact, there are quite a few tools. I can give you, hold on a minute, I can give you a few names of these tools so that you can search for them and see what's going on. For example, there's a tool to detect image issues called Image Twin, and there are other tools like Argos by Citility, which gives a risk score on the basis of the author's publication records or citations of already retracted research. And there's another one called Paper Mill Alarm and Signals. You okay?
Yeah, I would like to add one thing to the question of, I think that at least I work in the pharmacoepidemiology field, so don't know about preclinical studies and so on. But for what I see in pharmacoepidemiological science, the future direction is toward a more transparency of the studies, and so it means that publication of the protocol before study initiation, and as well as publication of scripts, for example, for statistical analysis. And there are also some, I think, journals that require a data availability statement that so you can check if there are open data, if you have the script, if you have the protocol, you can check every step and ensure that the data that you are provided is of good quality, and there are made also errors and so on. Don't talk about sophisticated. Okay, so yeah.
I think you also touched on a very interesting point, is the publicity around these drugs and the public opinion. Now, when the lecanemab, when the aducanumab was approved, I remember the big titles on newspapers by saying, the first drugs that actually modifies the course of Alzheimer's disease. Now, being a clinician, and I'm sure Marco shares this feeling as well, people were coming into the clinic and say, when can I get this? And so on. And they still do. They still come and say, why I've been denied this when this could be, I could have much more, because that's what the message that comes through. And this, in part, scientists and clinicians have a responsibility to put things right, but at the same time, we need to resist. And there is an enormous pressure on academics when we publish something to do press releases. Then they end up in the hands of journalists. You have no controls, and there are big titles coming up. So we need to be very careful on how we communicate our science as well, because integrity is not only in the way we publish science, but also in the way we communicate science.
Yes, integrity is about the whole research process.
Any more questions from the room or online? Yeah, one in the back.
Thank you. My question is, why do they approve drugs with so many side effects?
Well, it's a good question that I would probably, ask the FDA to begin with. And I think there were lots of people within the FDA community that actually resigned because they didn't agree to that process being expedited, or especially to approval to be given when we didn't know enough about the specific populations that could take the drug safely. But as I mentioned earlier, I think one of the main problems which led to approval in the United States was the amount of pressure that was put by the Alzheimer's associations and by patients' organization. But integrity comes also to drug approval. And in that particular case, there were lots of people in the committee that had very, very strong conflict of interests, which were highlighted by those in the committee who actually resigned. But nevertheless, the approval went ahead. It never happened, I think, to my memory, that an entire, the majority of the committee actually resigns after a decision has been made by the FDA. Does anybody remember any other cases? I don't remember any other cases. I'm older than anybody else here, probably. But, it is a good question. The answer is I don't know, but it would be a good question to ask.
Thank you.
Okay. Considering that the amyloid hypothesis doesn't explain so well all the complex pathophysiology of Alzheimer's disease, which one candidate do you consider as a potential alternative. I mean, which one is the best target in your opinion? I guess that is a complicated question and also multiple targets. But if you have $1 on which target?
Well, before I moved away from Sheffield, where I was before, we were involved in clinical trials of a tau target, and if I had $1, I would probably bet it on tau for a reason, because it's closer to what we know. It correlates with clinical symptoms. However, having said that, even treating that particular problem, I don't think is going to be the solution for Alzheimer's disease, as it has been for other forms of other conditions like cancer or some other conditions. I think is a condition which is multifactorial, and therefore thinking that one drug can solve the problem probably is not the solution. The other issue, which is a problem also in clinical trials, and I don't want to monopolize the round table, by the way, but I'll try and answer your question. One is patient selection for clinical trials. All this, we know that most of these processes, by the time they become clinically manifest, is probably too late to be tackled. Therefore, we should invest more in ways of detecting the disease at a much earlier stage. The blood-based biomarkers, once they will hopefully come to the clinic, will be probably of great help. But there are other aspects of detection of the disease which are not being paid enough attention to, simply because you think that you do an amyloid scan and the solution to the problem has been found, and the reality is not like that. And I think for the future we need to think about this disease as a multifactorial, multi-drug disease, which needs several drugs need to target the different pathophysiology processes.
Yeah, I fully agree. Unfortunately, we are involved in phase 2 trial, as you know, with bepranemab, that is an antibody anti-tau agent, and unfortunately failed. Just recently, the presentation was at the CTAD, and it failed. Primary endpoint on CDR, and it met the secondary endpoint on biological removal of tau pathology. But it's always the same question.
If this worked, it would be better. No, what I'm saying is these drugs do what it says on the book. They do remove tau, they do remove amyloid. The problem is, because probably that's not the only problem, you don't get a clinical benefit. And the other issue is when there is sufficient neurodegeneration already, does it matter really if you remove these pathological processes from the brain? Probably not, because as we know, we don't we only marginally regenerate neural cells. So that's a real problem.
We have time for one more question. Yes, second row. Mike is coming.
Grazie. So thank you so much. I was wondering it's your meaning of statistical significance. And I'm not just talking about the p hacking, right? I'm just, as a scientist, what is your meaning of statistical significance? So what is the meaning that you give to p-values? Yeah, exactly.
That you should never consider a p-value alone it's much more important to consider the effect size and the confidence interval.
Yeah, that is a big point, especially because you brought the example of...
I'm not, by the way, I'm not a statistician.
Yeah, I get it, but you don't have to be a statistician in order to understand that. That's my opinion. For instance, the T distribution was built on beers. It was a quality check for beers. And we use the same statistic to evaluate difference between biological effects. Well, you must be, in order to use inferential statistics, you must be extremely aware, and you have to have a really strong knowledge about the mechanism and the parameter that you are measuring. So there's this tendency of seeking for the 0.05, 0.01, which I think that has a story about a bias that Cox and Pearson didn't want to give over credit to Fisher, so they decided just to mention all of these two p-values, but there's a huge list of p-values with different purposes. But this is still strong. It is still strong in research. And I'm wondering, what is your opinion about it?
Well, the second line of that, the second bullet point of that slide, it says it all, isn't it? Having a statistical significance is equivalent to having a clinical benefit. No is the answer. And as Sally said, the effect size is important, as well as the actual everyday meaning of that statistical significance. The other thing which I've experienced in the past, I've been involved in some independent trials. One of the things which usually happens in these huge multi-center trials is that the data are given to an agnostic statistician. And that is a problem because, they come up, crunch the numbers with no knowledge, and they come up with a bunch of significant p-values. Then something which I didn't mention in my presentation, but for example, for the lecanemab study, what they actually did, rather than say the difference is 0.45, that was written in small print. In all the presentations which I've seen, they would say a 27% better results with the treated, in the treated patients compared with the placebo. But 27% of nothing is
still nothing.
Still nothing.
That's why you use confidence intervals. But it's the same thing. If you have a difference of 0.45, right? If it's statistically significant, it means that it's reproducible, because you will have a really small standard deviation whatsoever. And that is associated with the data integrity, because a lot of experiments are not reproducible. And so why is that? So we are really pushed to seek for p-values. But I think that nowadays we're facing a new perspective, which is more related to exploratory data analysis, where p-values are for, you cannot use that. And so instead of saying we have a difference of 23% or whatever, you still consider also things that are not significant from a statistical point of view, but they have a meaning from a biological, from the applicational point of view.
So I think that, for example, in pharmacovigilance or pharmacoepidemiology, sometimes we perform a reporting odds ratio, something like that, risk ratio, and so on. Sometimes we observe risk ratio, for example, 1.10, and it's significant. And you say, okay, this is significant. And sometimes you observe a risk of 2.5, and it is not significant. Sometimes you are more concerned about 2.5 because it means that you are not the sample, so you cannot identify the significance, but statistical significance, but it can be a clinical significance of this. So it's just numbers, but you need to interpret each number that you have.
Yeah.
Otherwise,
and I really appreciate that you brought the example of Altman, because plant Altman is.
I think this is a wonderful way to conclude this panel, this round table. I have another quote by Doug Altman. Statistics used to be seen as analyzing data, the combination of numbers. I've come to see that as the least important part. The difficult bit is how you design a study, collect the data, avoid bias, and provide an honest representation of what you found.
Thank you so much.
I think, unfortunately, our time is up and it's time for lunch. So thank you very much for staying behind, and thank you to all of the panelists for answering your questions.
Thank you to everyone. We will take a 1 hour lunch break. Thank you.